Okay, welcome everybody. Welcome back to the second session on the Darwin Tree of Life project. And in this session of talks, we're going to move on a little bit further and look at sequencing, assembly of sequences and analysis of data, and the speakers will give an update about where those are in different aspects of the project. So before we get started, can I also remind people that if you want to ask a question, it really helps to put the question in the box during the talk or towards the end of the talk. So it gives us, we don't have very much time in between different talks, so we can ask those questions as soon as we can. And please use the upvote system if you see a question that you really want answered. Okay, so let me introduce the first speaker in this session, which is Marcela Uliana da Silva. Marcela is a senior bioinformatician at the Sanger Institute working with the Darwin Tree of Life project. And Marcela is going to give us an update on one of the real challenges of these large-scale genome projects, which is how to assemble DNA sequence data optimally and with high throughput. So, Marcela.
So hi everyone, I am Marcela. I am a senior bioinformatician at the Sanger Institute. I work in the Darwin Tree of Life project. So I guess you know the Darwin Tree of Life project. We aim to sequence 60,000 species genome from Britain and Ireland. We are many. We are taxonomists, field scientists, wet lab scientists, bioinformaticians, evolutionary biologists, all together in this project to yield the best genomes possible for the study of molecular evolution and to yield data for conservation efforts. In terms of the data types that we have for this project, we have what we call long read data, such as PacBio, HiFi, and Nanopore, and we have what we call long range, which is basically short reads from Illumina, but linked like Chromium 10X reads or Hi-C reads that come from the 3D structure of the chromatin. So I work in the DToL, in the Tree of Life assembly team, and I will be talking on the perspective of the assembly. So our team has actively been testing the latest sequencing technologies, pipeline, and to yield the best pipeline possible for the different taxa that we are going to be sequencing. And I will show you our approach for the Lepidoptera at the moment, the moths and butterflies. So for this approach, we have three sequencing technologies: PacBio, HiFi, Chromium 10X, linked reads, and Hi-C from Arima or Qiagen. And we use this sequencing, this assembly pipeline here. So basically what we have is usually 25 to 30 times coverage of the PacBio HiFi reads for each of the species, and we use HiCanu or HiFi-Asam assemblers. Because the PacBio HiFi reads are so high quality, it's possible to separate the haplotypes even if we have low heterozygosity. So an assembler like HiCanu is going to produce an output of double the haploid genome size, because it separates the haplotypes. HiFi-Asam has a degree of purging, but then we separate the haplotypes with a software called purge dupes, which separates into primary and haplotigs. Then after that, we concatenate the primary and the haplotigs and map all of the long linked reads back to both of haplotypes. And then because they are linked, they will be pulled to their specific haplotypes. And we run one round of free-base consensus polishing this assembly once. After that, we then look for the mitochondria. So I have written Mito HiFi. It's available on GitHub. Basically, you just need a close related mitochondria species, and Mito HiFi is going to blast it against all of your contigs and is going to circularize it and annotate your mitochondria for you. Finally, after that, we take only the primary assembly and we do scaffold it to chromosomes with salsa using Arima or Qiagen Hi-C data. So I will show you a few results that we've got from this pipeline for Lepidoptera. So here you see Vanessa atalanta, the red admiral butterfly genome. I will walk you through the slide. So here in gray, you see the genome composition plotted, calculated with K-mares. You can do this with the PacBio HiFi or Illumina. Here you can see that the estimated genome size of Vanessa atalanta is 359 million base pairs. Heterozygosity is 1.1%. And in this case, we had 34 times coverage of the long read data. So here you see the result of that pipeline I just showed you. We have assembled 171 million base pairs into 210 scaffolds, where 50% of the genome are in scaffolds that are 12 million base pairs or longer. That's our scaffold N50. Here in this heat map, this is a high-C heat map. So basically in the diagonal here, all of these red squares here are the scaffolds with all of the high-C reads mapped back to them. So if you see signals off of the diagonal, such as here, in a heat map like this, it can mean different things. It can mean that the reads are mapped to, for example, sex chromosomes which share homology. So you're gonna find off diagonal for the sex chromosomes, but they also can represent errors in the automated pipeline. So basically here we see there are a few things that have to be curated because reads are mapping to one scaffold and to another. So this goes into our curation team and they do finalize the curation of this genome. As you can see here, MitoHiFi was also able to recover the mitogenome for Vanessa Atalanta. Here, a few more details on Vanessa Atalanta. This was assembled with HiCanoe. So as you can see from the primary and the haplotype stats here, we have basically recovered both haplotypes, which is great. We do analyze the quality of our genomes with a software called Mercury, which basically analyzes the k-mer composition of the 10x reads and the reads that are in the assembly. So as you can see here, we have the k-mers of the shared diploid part of this assembly, and in the heterozygous peak, you can see the k-mers for both of the genomes here, the primary and the haplotypes, which is great. So we have separated the genomes. Our QV is 57, which is really high, so that's really good. And finally, on the right-hand side here, you see the final high-C map after curation that was done by the amazing curation team of Kirsten Hova, and most of the butterflies that I will present here were done by Jonathan, the curation. And as you can see, we have nothing else out of the diagonal, and we have all of the more than 99% of the bases of this assembly are found in the 33 chromosomes, which matches the karyotype of Vanessa Atalanta. So this is a very high quality genome assembly. So the same thing here for the small white. The estimated genome size is 249 million base pairs. We have a high heterozygosity of 2.2% here. For this species, we have 55 times coverage of packed biodata. And here is the assembly result of that pipeline. So we have assembled 256 million base pairs in 45 scaffolds with a scaffold N50 of 10 megabases, 10.6 megabases. And here you see this pre-curation heat map basically shows us there is almost nothing to be curated. We can see the sex chromosome here, most likely. So if you look at the orthology analysis with Busco, so we find 98.5% of all of the orthologs are supposed to be in all of insecta found in this assembly. So it's pretty complete. The mitogenome was also recovered with MitoHiFi. Here you can see a few more details. So again, HiCanoe, and you can see the primary and the haplotypes well recovered. We can see the separation of the k-mers. And here you can see the final QV was 58. So just before the Chromium 10X linked reads polishing, we also had a very high QV already. So this is to be said about the hi-fi reads. You can do lots of analysis without polishing, but for now we have been polishing with Illumina once. After curation, this assembly also goes to the karyotype of 26 chromosomes, and we have 99.84% of all of the bases in the chromosome. So it's a very high quality assembly. So I'm just showing you here 2 assemblies, but we have many more, but we only have 12 minutes. So this is a plot, all of the scaffold lengths versus the genome length, sorry, versus the scaffold N50 for our Darwin Tree of Life assemblies. For the ones that have complete data, so they are the light blue here, and the ones that have only packed biodata so far, they are the dark blue. And we have also plotted the assemblies done by experts, and they are presented in the lab-based database, mainly done with short read sequencing and a lot of hard work. But as we can see, sequencing and assembly progresses, and we have a lot of very contiguous Lepidoptera assemblies here, which is amazing. So this really opens up the possibility for so much science to be done. So I'll give you one example here. We have the ringlet butterfly assembly, which is 28 chromosomes plus Z. And we have this mapped against the thought to be ancestor karyotype of all of the Lepidoptera, which is the diamondback moth. And if we have a look at the chromosome 7 here, it seems to be a fusion between the chromosome 20 and the 8. And the same we can see here in the 9, so between the chromosome 30 and 16. So basically, these genomes are really giving us resolution to study orthology with genes and also chromosome evolution, which is really, really nice. Right, so our assemblies are open to everybody. Here you see the red squirrel genome node. So our assemblies will be all available in rapid publications in the Welcome Open Research. So you can find, for example, the red squirrel there already. All of our data is available, so our partners, ourselves, and yourself can do analysis with it. So you can go to the GitHub entering page now. You're going to find all of this data that I just talked about and more. This is not updated as frequently because it's updated by us, the DToL team. If there is something there that you would like to see, you can always email us. But you should mostly look for these assemblies in the official databases like NCBI or European databases, because they will be there shortly. So, and they will be annotated, which is really good. The final considerations here for this talk then, I can say that PacBio Hi-Fi with one round of polishing and only one layer of scaffolding, which uses Hi-C, is yielding chromosome level assemblies for Lepidoptera. Of course, this won't be the same for every taxa, but that's what we have been seeing for Lepidoptera assemblies. They range from 200 megabases to a gigabase in size. The Tree of Life assembly team, the DToL team, is actively testing different data types in assembly pipeline. So if you would like to hear from us and understand what we have been doing, you can always write us. All of our DToL assemblies are freely available, so please use them. We are generating them so we can do a lot of science. And I have to say that we are hiring the DToL team, the assembly team from the Darwin Tree of Life, really needs software developers and bioinformaticians to join us in this very challenging and large project. So please, if you are a software developer, a bioinformatician that would like to work in genome assembly and analysis, please mainly write to Shane at the moment because the positions are not available in the darwintreeoflife.org yet, but we do have this position. So if you would like to work with us, we would be most welcome to send us your CV and a little bit about yourself. So I want to thank you for listening. There are so many people involved in this project. I need to thank all of them. And the amazing DToL team that I work on and all of the curation team, they are all experts and lovely people to work with. And I especially need to thank Shane McCarthy, my manager, with whom I discuss genome assembly in a daily basis. So thank you very much.
Thank you very much, Marcel. That's really, really interesting. There's quite a lot of questions coming in, and I'll have to pick a couple. One area that seems to be very interesting to a lot of people is, given the excellent quality of the assemblies, could you have a situation, do you find situations where you have a highly contiguous genome, but low busco completeness?
Interesting. I mean, not really. It's almost everything above 95%. But I would say that we should even revisit the busco genes because they have been, these orthologs have been decided with very short reads, fragmented genomes, right? So now it's time for us to revisit that. But yeah, I mean, given the busco is always very high, so we haven't been finding any low busco scores, no.
Yeah, okay, fantastic. Another question really is one about clarification, really, just to make clear that all reference genomes are necessary for your assembly steps, because for some groups there will be no reference genomes. But if I understand correctly, these are completely de novo assemblies that you're building.
Yeah, they are de novo, yes.
Okay. Also a lot of interest in what we might call contamination reads which don't come from the species that you're sequencing, but could be endosymbionts and the like, or parasites. Are you keeping those reads and also trying to assemble them?
Yeah, absolutely. So PacBio Hi-Fi gives us this unprecedented possibility of having, even if you have low coverage for certain cobions, we can assemble them with high quality. So we have found that actually a Wolbachia cobion of some of this Lepidoptera, and they will be assembled and they will be released too, yes.
Okay, thank you very much. I think we'll stop there and we'll move on to the next talk. Thank you very much,
Thank you.
Marcella. So our next speaker is Alexei Sokolov, and Alexei is project manager in the Data Coordination Center of the EMBL's European Bioinformatics Institute in Hinxton. And he's going to explain to us really this bottleneck of how these sorts of data can be managed and delivered, these large amounts of genome data for optimal use by the whole research community. So over to you, Alexei.
Hello everyone, my name is Alexei, and today I'm going to talk about integrated data trail live biodiversity genomics data management, analysis, and presentation at EMBL EBI. So the point for DToL Live data project is to provide high quality, comprehensive, and richly described genomes for eukaryotic species from Britain and Ireland. And from our side, the European Bioinformatics Institute is responsible for processing, annotation, and presentation of these genomic data sets. And the scale and complexity of the sequencing endeavor demands a number of connected informatics components, including different systems to support species and samples description, data validation, automated flow of data about samples and sequencing data from generators to databases, also systematic and rapid genome annotations, public data presentation, visualization, and species data tracking. So the main point of DToL Live data portal is to provide the community with a single access point to all the data from the DToL Live project from an intuitive web portal. Also to collect and provide a public end-to-end view of the processing status of any species that is a part of the project, and provide programmatic access through public APIs, filtering and searching, literature tracking, and data presentation. This is overall representation schema of data flow. It's quite complex. It has three major steps where we do collect information. So the first one is sample collection itself. We collect metadata about biosamples that will be submitted to ENA and biosamples. Next step is experiment itself. So it's a DNA, RNA integration step, library creation, sequencing, and metadata will also be collected here. And finally is the results of experiment. So it's row reads, map reads, assembled sequences, and so on. And this also will be submitted to ENA archive. So this is more detailed EBI data flow. So this part that with green boxes is data portal itself. Central component of the system is the Elasticsearch database, where we will store and collect all the metadata about different experiments. So again, samples, metadata will be submitted to biosamples through ENA, and also reads, assembled sequences will be submitted to ENA archives directly. And assembled sequences will be used by Ensemble to create annotation files. All these links will be collected in a central Elasticsearch database. Also, this database will serve as a API endpoint for tracking information that will be used by a data portal itself. And also we will provide public and RESTful API data and tracking endpoints. So this is our data model. So to study hierarchy, we have one umbrella study, it's a DToL Live, and then we have secondary umbrella studies for each species in particular, and for every species we'll have data attached studies with row rates, with the sample sequences, with, that will be used by Ensemble, and Ensemble will submit back files with annotation. So regarding submission to buy samples, the data model looks like this. We could have separate records for organism itself or for its specimens. So it might be liver, it might be brain, it might be something else. So we'll submit records for organism and separate records for every sample taken from this organism. And overall data model looks like this. So again, we have first one major study, umbrella study for down-to life, every study for every species, and then for every species we'll have data attached studies with experiments that will be connected to biosamples. So this is metadata about the samples itself. Also, experiment will be connected to runs, so with links to the actual files. And run will be connected to secondary analysis, including assemblage sequences and annotation analysis created by Ensemble. So now let's talk about data portal itself. On the front end part, we are using Angular framework and Angular materials for all functionality that we provide on a data portal. The main central part of a data portal is Elasticsearch database. As I showed earlier, it provides dynamic mappings for data and also provides rich capabilities for Google-like full text search. And between them, we have Java Spring Boot framework as a backend. It handles all requests, the Elasticsearch framework, and also provides API endpoints to front-end clients and users that wants programmatic access to data. Under the hood, system itself consists of a set of microservices that were deployed to Kubernetes cluster. And the system allows us to scale up and scale down automatically depending on the load. Also, system has capability to self-heal itself in case of any failures and provide convenient and seamless deployment without downtime, without visible downtime for users. First public release will be in October. This is the link that we're going to use, and this will be followed by periodic features releases. Now let's talk about data portal itself. So this is the entry point, homepage. Data portal has two paths. So first one is the data portal itself, just to explore all the data that are currently available inside Down to Life project. And also we have status tracking service that allows to explore statuses of every sample in a project. So you can find more information on about and help pages. For data tracking system, it provides high-level status of any species in Down to Life project. And we already set up initial set of full automated triggers points. So we have 5 main trigger points. So it will be metadata submitted to buy samples, raw data submitted to ENA, metrics submitted to ENA, and assemblies and annotations submitted to ENA. So for every sample, we have for every one of these 5 stages, we'll have 3 different statuses. So the status might be done. It means that for this particular point, the process is complete. Status might be waiting. It means that process started but not completed yet, and status might be undefined. It means that this point still needs to be done. For this status tracking system, we provide filters based on organism name, on common name and Latin name for organism. Also, we provide sorting capabilities through the table and Google-like searching. So it's possible to filter and find records that you're interested in. And for future, we will develop an API endpoints for receiving push tracking information. It will provide us with more fine-grained detail of processing. We will define JSON schema template for each type of trigger. And we'll have mandatory and optional additional metadata on the status update. For example, it might be metrics, quality scores, or heat maps. Second part of the portal is the data portal itself with all the information about collected samples. So again, we provide filters based on predefined filter components. It's possible to sort tables, it's possible to use full text search to find the records that the user is interested in. Also, for every record, we have a details page that provides full access to all the metadata collected for this sample. Provides information for related samples, and also provides information to all the related files. So it's possible to download all related files. It might be map reads, it might be row reads, it might be some analysis data. So it's possible to download it directly from data portal or through public API endpoints. For the future, we will provide Google style predictive search, hierarchical taxonomy search, navigate tree of life. We'll provide more finer grained status updates, additional status metadata information. We will do improvements on presentation and filter sites. Also, we'll provide embedded genome browsing, automated literature tracking, and whole page infographics and statistics for all the samples that are currently available for this project. So I'd like to say thank you to Peter Harrison. He's a data coordinator and presentation coordinator for his proposals, and also to Talal Ibrahim, who is our main developer for this data portal. That's it. Thank you.
Thank you very much, Alexei. Thank you. There's a lot of detail there that we all have to try and work with, but a huge amount of potential there. So what I'd really like to ask you to start with relate to questions that were coming up earlier in the day regarding people not necessarily working with the Darwin Tree of Life project really want to know what's the status of sequencing of particular species and where things are at. So I'm interested if you can say a little bit more about this status tracking feature that will appear on the data portal. Will that allow users to query for their favorite organism or indeed a particular taxonomic group?
Yes, exactly. So on this page, there will be filters. So it will be possible just to find your organism of interest using the filters on the left-hand side, or it's possible to do a search or sorting. And we will provide public API endpoints. So for those who want to get programmatic access, so it will be possible just to do this kind of search using the programming language.
And if related to that, at what stage does a sample that's been collected make its way to the data portal? Is it once sequencing has started, or is it once the species is collected?
So for now, all the statuses are quite, I would say, high level. So it's just metadata about the sample was submitted to biosamples, and row reads was submitted to ENA and so on. But we are working on push tracking system, so it will be more granular control over the statuses that we have. So it will be like the sample was collected, DNA was extracted, and so on.
Right. Okay. Fantastic. And considering that many of us are used to working with NCBI bioprojects, I noticed in your links and arrows that the bioproject is there, and of course all these samples will have a bioproject. But if users were searching in bioproject, they would be able to find their way to a Darwin Tree of Life data portal, would they?
Using just EBI, it won't be possible because it will be just an ID and information, but from the data portal it will be possible to find all the IDs and go to the archives itself.
Right. Okay, great. Thank you very much. I think we'll stop there for questions and move on. Thank you very much for giving us that insight into the data management. So now we move on to 3 shorter talks, and the first one is from Alex Twyford, who you've already seen as he's the co-chair of the Darwin Tree of Life sessions at this meeting. Alex is a NERC Independent Research Fellow at the University of Edinburgh, and he's going to be talking about sequencing of plant genomes. Over to you, Alex.
The Darwin Tree of Life project has the visionary goal of sequencing the genomes of all British and Irish organisms. And today I want to tell you about what we can expect to learn from the plants. And perhaps the key feature we have in terms of studying the plants is that we know a huge amount about the British flora. We know lots about the ecology and the evolution of all the species, including chromosome counts, information on plant communities, and information about hybridization. And all of this is a powerful context to place our genomic data into. Today, I want to think through what we can learn from broad-scale analyses of plant diversity, so why it is that we want to sample all of the plant species in Britain and Ireland. And I'll give an example of that using DNA barcoding from our work so far. And then consider why it is we need whole genome data to understand the evolution of the British flora. And I'll end with thinking about how the Darwin Tree of Life project falls between these two areas and covers both breadth and depth of genomic data. So in terms of broad scale work, we've recently completed the DNA barcoding of all British native plant species. And that includes 3 loci for nearly 1,500 species. And to place this in an ecological context and to integrate our genetic data with ecological information, we've compared this genetic data to information on hybridization. This is based on Clive Stace's hybrid flora, which tells us about all British native plant species and which species hybridize. Based on this, we've completed the most comprehensive phylogeny for the British flora. And then in green here, you can see the prevalence of hybridization between each pair of taxa. And then in blue, we've got a model-based analysis incorporating information on chromosome number, genetic diversity, and a range of other factors. And this shows that certain groups are more prone to hybridization than others. But what's really critical is based on this analysis, we can work out the main factors driving hybridization across all species in the flora. And when we do this, perhaps the most important factor is genetic distance between parental progenitors. In this figure, I'm showing separately for ITS and for plastid data, the genetic divergence between parental progenitors for those species that do not hybridize and those that do. And what you'll see here is that those species that do hybridize have a much lower genetic distance. And that clearly emerges the main factor affecting the prevalence of hybridization. But DNA barcoding data is not sufficient, and we need to move on to generating whole genome data to really understand evolutionary processes across the flora. And here I want to give you an example from my work on British eyebrights or euphrasia, which are a group of 21 species, including diploid and tetraploid species. We've sequenced the genomes of 18 different species, and using these data, we can identify the genomic scaffolds that are shared between diploids and tetraploids, and those that are unique to the tetraploids. But to understand the divergence and diversity of diploids and tetraploids, we've also had to develop new analytical tools. And the primary one we've worked on is called TETMA. It's a KEMA-based tool that looks at the KEMA spectra, so the frequency of each different short sequence. And we can use models that distinguish between diploids and tetraploids and classify the tetraploids as auto or allotetraploids. When we apply this to the tetraploid genome data here in the top right, you'll see that all of these species have very similar subgenome divergence, telling us that they're probably evolved from a common ancestor and involve similar parental progenitors. What will we eventually learn from having whole genome data for all species in the flora? Well, I've written about this in this Nature Plants commentary paper and how we'll learn about phylogeny and ecology and all sorts of factors like horizontal gene transfer. And I think our biggest barriers to this are getting high molecular weight DNA extraction optimized, sorting out the assembly of large and complex plant genomes, and really understanding comparative genomics and getting these new tools developed. So I'd like to end by thanking the very many partners involved in Darwin Tree of Life and all the people who've contributed to this work. Thank you.
Thank you very much, Alex. So you've given us there quite a focused view on hybridization, obviously something really key in the evolution of plants. But I was also wondering about whether you think you'll be able to use whole genome data when we only have single representatives from different species to find genetic basis of adaptation in plants?
Yes, so I think we will, though we'd have to temper that expectation with some limitations there. I think that by doing broad scale comparisons by habitat and then by group, we'll be able to find, for example, loci involved in adaptation to montane environments or harsh environments or high areas, though I really think, and we'll also be able to get some of the pathways underlying that. But I do think we really need to get resequencing data as well to really look at genetic diversity within species. So we need to link within to between species.
Yeah, I think that's probably something that we should probably all emphasize as part of the Darwin Tree of Life project, in making reference genomes available should hopefully stimulate research groups to do resequencing and address particular biological questions through their own resequencing efforts. I mean, do you have particular lines of resequencing work that you would be interested to do in that direction, Alex?
Yes, well, I'd like to resequence everything, as we all would, but realistically, I suppose we would probably focus on closely related taxa, hybridizing species, and really try and guess at whether introgression is fixed within species or geographically restricted, and whether there's certain loci underlying adaptation. That'd be a particularly interesting avenue of research.
Yeah. Okay, thank you very much. I think in the interest of time, we'll move on again. There may be time at the end for more questions, but we'll see how timing goes. So thanks again. So our next talk is going to be given by Peter Mulhare, who's a postdoc at the University of Oxford, and he's going to be talking about recent findings with some of the draft genome data coming out from Darwin Tree of Life, particularly on insects. So over to you, Peter.
Hi, thank you very much. Today I'm going to be talking about our analysis looking at homeobox gene evolution across Lepidoptera in collaboration with the Darwin Tree of Life project at Oxford. So homeobox genes, we know, encode transcription factors that are crucial for the regulation of development. They've been annotated in a cluster of genes and are important to tell cells where to go during development. However, we know that the Hox cluster, images of the Hox cluster are often incomplete, and there's quite a bit of a divergence between species and between phyla. For example, in Lepidoptera, we see the emergence of 4 SHX genes within the cluster, and even some more highly divergent Hox genes within certain species. So to look at homeobox gene evolution across Lepidoptera, we use data that was available from the Darwin Tree of Life project. The benefit of using this data is this broad sampling of species that they have, but also the fact that they're sampling really closely related species. And in doing so, we can get a really in-depth insight into how homeobox genes are evolving. To look at these gene families, we used 36 draft genomes that were available to us. So first, we looked at the Hox subclass of homeobox genes. We're able to annotate these genes for the 36 Lepidopteran genomes available. And the first thing to say is kind of the quality of these genomes is really quite high. And we're able to annotate most Hox genes on usually on a single cluster or a single config. Constructing a phylogenetic tree, we're able to group most genes into their families, and we're also able to confirm these fast evolving rates of the SHX genes. We're also able to annotate a kind of normal Hox cluster within Lepidoptera. So for example, in the silver Y moth, we see an inversion of labial from to the opposite end of the Hox cluster. We're able to confirm the presence of the 4 SHX genes within the cluster. And in all species, we see the introduction of the rough homeobox gene, which is usually associated with a different subclass of homeobox genes. Possibly the most interesting finding is these dramatic expansions within the Hox cluster that we find in certain moth lineages. We're able to confirm that these are biologically real events because we have this deep sampling. So for example, in the 2 sister Blastobasis species, we see a large expansion in both of these Hox clusters, suggesting that it's not just an artifact of assembly and it's actually a biologically real event. Taking the example of the ring China mark moth, we wanted to see whether what possible function that these expansions could have. What's interesting about this particular species is that its larvae have an aquatic stage. And we know from previous analysis that the SHX genes are involved in egg development. So this gives some idea, potential role for these expansions in the adaptation to an aquatic stage. Next, we want to look at the NK subclass of homeobox genes. And what we find is a kind of a different view of how homeobox genes evolve within Lepidoptera. So we see much less divergence and fewer changes within the cluster across all Lepidopteran species. We were also able to annotate the canonical NK cluster and find that relative to other insect species, we see some genes being removed from the NK cluster. It's also a useful control against the Hox cluster, as we see that some of the species that have really highly divergent Hox genes have more conserved cluster of NK genes, again suggesting that these changes in the Hox cluster are not due to any assembly error and are biologically real. When we construct a phylogenetic tree, we see these more conserved rates of evolution in the NK gene families relative to the Hox genes. Suggesting a different role and a different way in which these homeobox genes are evolving. But we know in the Darwin Tree of Life project that there's sampling of species, arthropod species outside of Lepidoptera. So what about the homeobox genes outside of Lepidoptera? So what was available to us at the time was 4 hoverfly genome assemblies. And interestingly, we find in some of these species similar large tandem duplications within the Hox cluster. So the same expansions we see in Lepidoptera, we also find in hoverflies, suggesting an adaptive role for these large expansions within the Hox cluster. So to conclude, it's undoubtedly deep sampling and high quality genomes really provide key insights in how gene families evolve. For the Hox cluster of homeobox genes, we're able to confirm the presence of the 4 SHX genes within Lepidoptera, and also find some interesting patterns of gene evolution, such as these large expansions we see in certain moth lineages. We see a different view of the NK cluster of homeobox genes of a more conserved evolution relative to Hox genes. It also highlights the different ways in which homeobox genes may evolve. So I just want to say thank you for our collaborators and for all the funding that was made available to us for this analysis. Thank you.
Thank you very much, Peter. So the first question that's come in relates to prediction pipelines, annotation pipelines, if you like, for finding Hox genes and other homeobox genes in the genome data. So the question is whether you have developed a dedicated method for doing that, or whether you're relying on annotations.
Yeah, so because there's no annotation data released for these genomes yet, we've had to go in and search for these genes ourselves. And to do so, particularly for the homeobox genes, because they contain a homeodomain sequence of 6 amino acids within the protein, we're able to search for that using a BLAST and a best reciprocal BLAST hit. And so then we could get really a good idea of what's actually present and how they're arranged. And we do that in kind of an automated fashion.
Great, so that's a pipeline that could be applied to any genome sequence. Yeah.
Exactly, yeah, exactly.
I also have a question which is a sort of a taxing biological question which puzzles me. When we look at those Hox gene structures that you showed for different moth species, they sort of fall into 2 categories. Either everything's normal and not a lot's changing, or they just go absolutely crazy. There didn't seem to be anything in between. Do you have an insight into that?
I don't really. There was a couple that had maybe a single duplication of a SHX gene. I think only one other. But as to why there's such huge divergences versus conservation conservation within the host stressor, I really don't know. We're not even sure what these expansions are doing themselves. So I'm not sure why there's this difference, huge difference between species.
We'll squeeze in one more quick question, which is about hoverflies. It says, why does having more SHX genes help aquatic larvae? If SHX genes help aquatic larvae, why do hoverflies have expansions but not aquatic larvae? Now, as I understand, some hoverflies do have aquatic larvae, but I don't know whether that's the case for the species that you've got the expansions in.
Yes, yeah, you're right. There is one, aquatic hoverfly that someone else has analysed that has this large expansion. But in the ones that we have, I don't think they're aquatic. But I guess the aquatic stage is kind of just one hypothesis as to why you would see these expansions, and in hoverflies there could be all sorts of reasons why we see it.
Great. So I think that emphasizes another point about the Darwin Tree of Life project, which is that the genome is the start of biology. The biology starts from here on. Thank you very much. So we'll move on to our last talk of this session. And this will be given by Alan Tracy, who is a senior computer biologist at the Sanger Institute. And he's going to be talking about analysis of the genome of a very important pollinator species sequenced as part of the Darwin Tree of Life project. So over to you, Alan.
So I just want to show you some points of interest from manual curation of Ceratopipians, and then briefly discuss some insights into Diptera and chromosome evolution. The genome was assembled using the DToL pipeline, which involves PacBio Hi-Fi, polishing and Hi-C scaffolding. On the left you can see a Hi-C image of the final assembly after curation. You can also see that the total assembly length is very close to the estimated size. A small unresolved repetitive scaffold leaves total scaffold count at 6. After automated assembly, a round of curation using Hi-C brought the number of scaffolds down from 20 to 7. From there it became less obvious how to make the remaining 2 joins to achieve the 5 chromosomes displayed here in the final Hi-C image. This is what I want to take a couple of slides to explain. As an aside, manual curation involved resolving a 3 megabase inverted duplication introduced at the high canoe assembly stage of the pipeline. Here you can see that the first 3 megabases of this chromosomal scaffold consists of an artifactal duplication which has now been removed. To look for further joins, I made a protein comparison of Drosophila on the right against Ceratopipians on the left, with the connections coloured by Drosophila. It's clear to see that scaffolds S1 and S4 are strong candidates for joining, as they both come from the same Drosophila chromosome arm, coloured red. Going back to the Hi-C map, I could see a weak but significant Hi-C signal between S1 left and S4 right, and these scaffold ends were found to have a shared large tandem repeat. This repeat gives us an explanation for the weaker than normal Hi-C signal between these scaffolds and a mechanism by which they would join. So we know that one more join is needed. According to synteny evidence, there are 5 possible joins involving 3 pairs of scaffolds. Either end of S6 to the left end of S3, the left end of S3 to the right end of S2, or the right end of S2 to either end of S6. Here's a plot produced by a tool in development at Sanger called Genome Decomposition Analysis, or GDA. One plot it produced was a summary of repeat distribution across the genome. This allowed me to see one particular classified repeat type, highlighted here in red, which occurred at the left ends of S3 and S6. These ends happen to be one of the 3 choices available from synteny evidence. The sheer size of this 2 megabase tandem repeat gives us a good explanation for the missing Hi-C signal that was preventing us finding the join in the first place. Furthermore, if we compare the relative sizings from boys with relative sizings from each of the 3 join possibilities given by synteny evidence, we can see that S6 to S3 gives the best fit, an additional piece of supporting evidence. Pablo has run some analysis of busco genes linked to Drosophila muller elements in Diptera, including the new S. pipiens assembly. From this, we can draw 2 broad conclusions about chromosome evolution in Diptera. Firstly, Ceratopipians gives clear support for the division of Muller-C into two elements that reside on different chromosomes. This is interesting because it's accepted that Muller elements broadly represent dipteran arms. This assembly is telling us that this is not the case for Muller-C. Given that in Drosophila C is found as a single unit, there are two explanations for this. Either the ancestor of dipterans had C as a single unit and then it was split in the ancestor of mosquitoes, or it was found as two units and only in the ancestor of Drosophilids did these units fuse together. Secondly, Muller-B has been conserved as a chromosomal arm in all dipteran chromosomal assemblies so far. This is the first assembly to show the clear breakage of Muller-B, which must be a new rather than an ancestral characteristic, as B is found as a single unit in both Drosophila and mosquitoes. In conclusion, Ceratopipians is the closest chromosome-resolved assembly to Drosophila and gives us insights into dipteran chromosome evolution, specifically showing that Muller-C can be further subdivided. The breakage of Muller-B suggests that translocations are not restricted to chromosomal arms. Some detective work was necessary beyond looking at high-C data to resolve this assembly into chromosomes, and GDA was an important part of this process, helping to give greater confidence in identifying a sex chromosome in this homogametic sample and helping us find large shared repeats at chromosome ends to narrow down our joined candidates. And I'd just like to thank all the people at Sanger who have been involved in helping me with this analysis. Thank you very much.
Thank you very much, Alan. So my first question really, I'd really like to ask you about the generality of the type of analysis that you needed to do to really refine. I mean, you've done something quite remarkable there to refine that assembly to that level of contiguity. Do you think that's going to be needed in several other of the assemblies that come out from the project?
Well, I find that I often have to do a lot of investigation in order to refine an assembly that's automatically produced. There's often a lot of clues, and it involves looking at a lot of different, all available data types. You know, there might be high C, might be GC plots, might be all sorts of things.
Yeah, so that's something to bear. I think that's probably something we all have to bear in mind when we, and of course there's a payoff between the work that's put in and what comes out, and that's going to be different for different species, I imagine. That leads me to ask why this particular species. I mean, I was intrigued, of course, that this particular hoverfly is an important pollinator species, but I was wondering why you chose to study this species and the genome of this species. Is there something particular about chromosome evolution in that lineage?
This one just landed in my in-tray, and I always like to take an assembly as as far as is feasibly possible.
Yeah, great. There's comments coming in, one of which is very nice. I think people are going to have lots more to explore in terms of synteny and genome architecture now with these chromosome level assemblies. I think that's right. This sort of opens up a whole new area of biology that we couldn't really start to address before we had this level of genome information. So the future is bright ahead. So I think we're going to have to stop there in the interest of time. I want to do two things. First of all, to thank all the speakers, both in this session that we've just had and of course in the previous Darwin Tree of Life session. Thank people for listening as well. And also remind you that there is a poster session, a virtual poster session, which we can now move to. And I believe it is in this space site where you appear as a little icon walking around a room. So I hope to see some of you there. Okay, well, thank you again.